So good afternoon, everybody. It's now quite a good daunting task to start talking about Ian, and I will give the example of whether I have actually understood your marvelous research because I have a slide of your work, and so we'll go to that. First of all, I would like to say thank you for inviting me. This has been terrific 2 days, and it's a real pleasure to be here to talk about hallmarks of environmental insult. And with the representatives, environmental exposures impair health globally. And we have these overarching issues, which everybody has also voiced. We have the climate crisis. We have aging populations. We had the corona pandemic, which was unexpected in its vastness. And we have increases in social disparities. And Claire has explained to you this morning, very beautiful, how cohort studies have kind of really nailed down ambient air pollution. And to my mind, for PM, for fine particles, we are in a way done. We have so much evidence. We know that this is harmful. It's inducing mortality. And so we would not need another cohort study for that. But this is the world of cohort studies, how we're conducting them today. So we look at lifetime exposures. We characterize the plethora of issues, environmental, food, smoking, exercise, medications, and so forth. And we are really getting good at that. We are developing more and more tools to do that. We are also have been advancing as scientists as a whole our understanding of biological responses. The genetic makeup, which kind of contributes to disease, the epigenome, so the regulatory processes, all the molecular things we can do today, and which we're doing and including in our cohort studies. And then we are investing into sophisticated phenotyping of us, basically. So we are imaged, we are measured. Our 3D echocardiogram is taken. We take biosamples. And so we really invest heavily in understanding the pathways from disease, from environmental exposures down to early signs of disease and then manifest diseases. But again, as said before this morning by Claire, we know fine particles lead to death. So the question is, how can we be more intelligent in understanding the impact of environmental exposures? Because to my mind, here everybody is convinced that environmental exposures are harmful. But if we go to our medical colleagues or to those who do experimental work in understanding the diseases we care about, they don't care about environment. They care about genetics. They care about targets. So in a way, I think one idea could be to actually go to the plethora of exposures and look at what do they do at the cellular level. So when basically these environmental exposures, which are all around us, kind of hit our body, whether it's within the lung or within the blood, but what is happening there? And I think one of the challenges we have is that the environmental exposures come in so different flavors. We have the particles, and we discussed the microplastic particles, but there are these plethora of chemicals, as we have seen in water. But there is radiation, which is a completely different physical exposure. There's noise, which we hear with our eyes, and somehow we need to have concepts which jointly characterize this if we think of modern epidemiological studies. And so the idea is to think about the health impact of these things jointly, because this is also where we are moving in exposure assessment. And to think about molecular and at the cellular level, how does it affect our organs, to be able to also talk to our science colleagues. And so Tim, Andrea, and myself, we had the idea that we would conceptualize this further. And we started with an analogy. Namely, there is a paper in 2013 which looked at the hallmarks of aging. So explaining biologically how we age and what are the molecular mechanisms in our body which lead to the changes as we have seen this in Ian before. And we thought that if environmental exposures really matter to the degree we think, they should also be driving these aging processes in addition to leading to specific diseases. And so if you look at the aging, there's cellular senescence, there is maybe deregulated nutrient sensing, but there's also other markers of actually genetic deterioration, changes in the control how our cells are functioning, which is kind of getting worse as we age. So the idea was to really take this and review where do we have evidence that environmental exposures are relevant. And it turns out there is a really very nice overlap. We have genetic alterations. So there is an accumulation of genetic degeneration with environmental exposures. Actually, the gene function changes. The energy of our cells is impaired, so the cell cannot work as well anymore with environmental exposures. There is endocrine disruption, meaning that our hormone regulation is not working well with environmental exposures. And there is our body relies, our whole functioning relies that the cells and the organs communicate well with each other. And there is actually, this is impaired both in aging but also with environmental exposures. Then we have now more and more evidence that there are altered microbiome communities. So this idea that we are many more than just us here, and that this is really impaired by environmental exposures. And then there is something we have actually also impaired nervous function. So if you hear loud noises, it's kind of, the first recipient is the ears and your brain. So, or you breathe in particles and there's a change in autonomic nervous system, or you sense heat. So again, so this is something which is actually not implicated in aging, but I think is very relevant for environmental exposures. And then if you just take air pollution, one can see, well, one of the critical things is inflammation and oxidative stress. Air pollution brings, or particles bring into our body organic chemicals which adhere to our DNA. We have shown that there are changes in the epigenetic regulation, and we can actually have a clock which shows how these changes accumulate in us. There is work on mitochondrial dysfunction, and that is something Tim has shown, especially for in the placenta as well as in blood cord. You can show that actually the energy supply for the cells is already impaired. There's a few papers on hormone dysregulation. There is actually evidence on senescence, so aging-associated phenotypes, and altered cell-to-cell communication. There is reduced microbiome biodiversity in the gut in relation to air pollution exposures. And there's altered autonomous nervous system and direct impact on the brain by particles. So I will now kind of give a little bit more meat to this overall concept and start off with the description of the particles, where I think it's very important to see that this is always a mixture, and that we have all the ingredients on the particles, and we often have, if we care about the diesel particles, this carbonaceous core in the middle. And so one of the so beautiful pictures is this, because air pollutants actually induce oxidative stress. So, and air pollutant exposures reduce our antioxidant defenses. So the important message is that all our cells are actually protected. And the reason why they have this protection for oxidative stress for radicals is because the cell uses these radicals for regulation. Everything in the cell, which is kind of all the things that it function in, is using oxygen radicals. So that's why it needs to be very protected. It's only if you get too much pollutant that it starts to ramp up defense mechanisms. And it has actually two. It starts on with something which is still adaptive, and then it starts to really say, hey, I need help. So then there are these, the cytokines which are released, and as a consequence, the cells actually may be destroyed, and then you have the long-term impacts. So I think this is really a beautiful concept, how the particles impact the lung at the first interface. And this is really your work, having worked this out, how this goes. One thing is you can use these understanding to understand, for example, desert dust. And to say, well, if one reviews the evidence, as you have done, this is a risk factor for inflammatory and allergic lung diseases. So really saying, well, this is what has happening here. And it's not the it's the adhered chemicals up here, the biological and the mineralogical components which are candidate activators. And it's the surface reaction, which it may enhance toxicity of aerosols in urban environments. So in a way, the understanding what we have, together with characterizing these dust particles, we're done. We know that these particles need to be still controlled, and that we cannot discount them if we have the new air quality guidelines. So we do not, EPI is more helpful here, but I think we have actually sufficient evidence to have clear messages. And the next thing which I find important and has really been worked out in this interplay of epidemiology and toxicology is these local and systemic health effects of air pollution. Because when I started in '93, I worked on asthma and particles. So it was clear that there is a relationship. But the link, which then came up in the next years to be the link to heart disease, was actually used as a counter-argument that particles could be harmful, because everybody understood the lung is the target. But if you also see your statistical association with heart disease, this is a clear sign that correlations are wrong, because there was no mechanism. And again, out of these combinations, we have actually now have evidence that here you have the inflammation in the lung, which then leads to systemic oxidative stress and inflammation. We have the particles which are translocated into the bloodstream, again, something which we only can find out by toxicology and not by epidemiology. And we have an activation of the autonomic nervous system. And one of the amazing things which I was involved in, and a study funded by the HAI, was that we actually found that being in traffic increases the risk of having a heart attack 3-fold within the next hour. And it was regardless whether one was sitting in a car, in a bus, or on a bicycle. And it was very robust, and due to controlled exposures, this could be corroborated. So the studies which were done in Umeå, looking at exposing persons with chronic artery disease, so people who are prone to get a myocardial infarction, to diesel particles, which were ultra-fine, but relatively high concentrated. But this is what you have in tunnels. So this is not out of the range. And you see the heart rate goes up when they were exposed because they were exercising. However, if you have clean air, there were signs of ischemia, so that the coronary arteries were constricting. However, if they at the same time were breathing diesel particles, this was much stronger. And so this is a sign, not quite a myocardial infarction, but close by. So again, it's this interplay which really made us move forward and made us understand that certain things with the particles are very rapid, within minutes or within an hour. And then there are responses which take a little longer, and then you have the inflammatory responses which are even longer. And to my mind, we had this debate just about the short-term versus the long-term effect. If you take forward a pathophysiological understanding, having these insults over and over again, you can actually quite easily conceive that over periods of time, this accumulates and leads to atherosclerosis, so calcification of our coronary arteries, which then is a requisite that a myocardial infarction occurs. So I think you can actually infer, when you look at the mechanisms, and if it's only short-term, you can infer for the long term if you have the epi. Without epi, of course not, but if you do, you can. And so one of the cool things, which is like around the block now, is further understanding this crosstalk about organs. And so this was actually coming out of paired studies where Valentina Bolatti at the time looked at the release of cells of vesicles, of messengers, which would be consistent with cell-to-cell communication. And she also was able to corroborate these findings in policemen who were on traffic duty before, when they started, and after they had these messenger molecules mounted. So that we have there now Andrea Baccarelli in Colombia is following this up and saying, well, how does it work that after exposure in the lung, we have really this systemic impact, and it's small vesicle which now carry these messengers to other organs and can, well, first of all, they cry for help. But if this is overrun, then disease can develop. And actually, going back in time, 1985 was the last air pollution episode we had in Central Europe. And this is data from Augsburg, where the Accord study was started at the time. And we had actually measures of plasma viscosity. And when one compared the period at these high concentration with other times, we were able to see that plasma viscosity, so the thickness of blood, was increasing during the episode, as well as at the time we had sulfur dioxide, high sulfur dioxide concentration, and high total suspended particles. As medical science improved, we have a C-reactive protein, which is a marker of an acute phase inflammatory response, and we were able to show it's actually, we get even larger risk if we measure C-reactive protein. But just when I put this together for this talk, and thinking again about the paradigm of this intracellular communication, maybe the plasma viscosity was not just the inflammation, but also part of this massive response of our lungs towards the particles. And one other area where I think experimental work is really paving the way is coming from work of a colleague of mine, where he asked the question whether nanoparticles can activate dormant herpes viruses. So what he does, he infected mice with herpes virus, and this is the positive control. So the virus is emitting the infection in the mice if they are sacrificed after 6 days. Well, after 29 days, actually the infection is gone. So the herpes virus has internalized itself into the lung cells. It cannot be detected anymore. If one adds carbon nanoparticles to it, actually the virus reemerges. And if there are double-walled carbon nanotubes, so nanoparticle fibers, they also reemerge. And so this is something which we would not have been able to pick up with epidemiology at all. And if you consider this interaction with the corona pandemic and other viruses, this may be really important. And it's actually interesting because it's a different mechanisms of activation than inflammation. It has nothing to do with the normal response. And we started to look in human data, and maybe we can corroborate these findings using antibodies. So again, this would be then in my hallmarks, it would be this impact on altered microorganisms in this framework. And so just to final start, the emerging evidence we have for particles affecting the brain, and ultrafine particles playing a major role here. Again, this is work which has been worked out with animals. It's the inhalation of particles. The ultrafines deposit in the upper airways, and they are translocated via the olfactory nerve to the brain. But they also go into the lung, where they're deposited. And, well, actually, if they land here in our upper airways, we swallow them. So they go into, or they're in the upper airway, and they go into the gut. So then toxicology has shown us that actually from the ultrafine particles and constituents on the particles can leave the lung, they enter the bloodstream, and they actually do the same thing from the gut. Then they actually passage the heart, go once through the lung, and then up into the brain. So that we have actually the passage of these ultrafine particles and their constituents all around our body, very consistent with impacts on the brain, and where they then can impact, again, diffuse inflammatory responses, can contribute to cell misfolding, which is the hallmark of Alzheimer's disease, and can also lead to vascular dysfunction and potentially stroke. So we have all this evidence, and we can also talk to our more science-oriented colleagues and say, well, look, with the knowledge we have really impacts on the organ level, which is in line with modern ideas about organ aging as well as disease development in these organs. So to my mind, toxicology shapes the future jointly with population-based studies. And just to say, I think it's essential for understanding of health effect that we target, ask really smart questions. And I was specifically impressed with the framework which is now available for adverse outcome pathways. So how that a certain exposure can be linked to a specific disease, such as, for example, lung fibrosis, and that by characterizing different particles which one may find in occupational settings or in the environment, and I was also thinking of potentially the fibers, the plastic fibers. If one can then show dedicated pathways, then I think one does not need epi. One can simply go right away and saying this fiber is a problem and this is not. And then I think the potential to identify novel hazards and novel outcomes, which I think is important and can be done from both sides. And so to just sum up, we have actually heard here the excellent work you have been doing on changes in particle toxicity with the implemented mitigation measures. So I think we will be hopefully seeing the changes to get clean air and mitigate climate change. And so the particles will be changing, and we need to understand that they do this in the right way. There is a non-tailpipe emissions and the wonderful work on microplastic. And it's also very important that we quantify the benefits of intervention holistically. And luckily, I did not have to think of these important points myself, but was simply taking this from this very nice review. So congratulations to 30 years of laboratory and field toxicology. That's how Bob Devlin from EPA calls epidemiology, and good luck for the next 30 years. Thank you very much.
Thanks for a really fabulous talk. We've got some questions. Maybe oh, there's one up there. Okay, I was going to start, but yeah.
Thank you, Annette. Just beautiful talk as always. And your case crossover study on traffic is one that I cite the most, probably of all studies. So very exciting to see you show it here. And actually, my question is related to that. So I've done quite a bit of research on physical activity and air pollution, trying to understand the risks and benefits. But despite my having done a lot of work on this, I still don't really understand. And I thought you'd be the perfect person to try to explain what you think the mechanisms are. So when you cycle and you cycle habitually, when you're physically active, you have reduced systemic inflammation, right? And that kind of counterbalances the air pollution impacts. And in your study, you saw that even people who were cyclists had the risk of stroke or ischemic heart disease or whatever it was. So I was wondering if you can explain what do you think that mechanism between the physical activity, like from your perspective, I would love to understand what happens.
Mm-hmm. So this is a study where we looked at myocardial infarction survivors only, and you asked the question, is what do activities shortly before the onset of the symptoms are different compared to like the 4 days before. And this is because the underlying pathology of a myocardial infarction is that you have a vulnerable plaque in your heart, in your coronary artery disease, and that at some point it ruptures. And there are triggers which can do this. And exercise is one of these triggers. So it's basically the increased heart rate and the constriction. So if you cycle hard, you need to have the blood flow through your body. And this can kind of tick off such an atherosclerotic lesion. However, as a trained person, you need to have a vulnerable plaque. So as a trained person, actually you work against that. Even if, for example, if you have genetically high cholesterol levels, but are exercising a lot, you still get the plaques, but the plaques are more stable. Whereas somebody who is not exercising has a vulnerable plaque, and when then this person exercises, this may rupture. And it seems that also when we breathe air pollution, we have an activation of the autonomic nervous system, and that can also lead to constriction of this vulnerable site. And so you have these transient risks, and you have the either increased by air pollution or reduced long-term risks through physical activity. And we also in the same study looked at physical activity and can beautifully show that. So physical activity is an increased risk, but it's more so in people who have sedentary lifestyle. And those who are active, they actually have a much less likely chance to have a myocardial infarction. And again, I mean, this triggering is really transient. So I think the concept is that we have these overlay of time mechanisms, certain things which kind of accelerate our aging, and then other things which make it more likely at this moment in time we conceive an acute event.
Oh, there's one here.
Yeah, thank you, Annette. This is fascinating, and it's a good bridge between our field epidemiology and field toxicology, and toxicology. Nowadays there are a lot of research on air pollution and mental disorders, in particular depression, anxiety. And we have been struggling with toxicologists about the mechanism for this. You showed this nice path for ultrafine particles, but you were mentioning basically neurological disorders and stroke. Do you think this pathway could apply to mental disorders too?
Yes, so I would say yes, but I think indeed this is an area where we do not have sufficient knowledge. So you can, for example, just out of the when you look at biological markers of mental health or anxiety, inflammation comes up. But it's not clear that in these settings it's really more a bystander. Or maybe it has a more crucial role. But I think there we really would need to have the right intelligent experiments or the mechanistic understanding which brings that together.
So one last question from Ian.
I didn't mention the exposome at all in my presentation, but I was thinking yesterday when we were talking about the social aspects and around this topic, that actually there's lots of evidence now that social deprivation, early life trauma itself, triggers accelerated aging. And so I was suddenly thinking, should we be considering poverty as a poison onto which air pollution is superimposed?
I think, yes. So I think this is again, I think there we probably need to think clearly also from a mechanistic side what we think is coming first and what is second and what is actually an enhancer of an underlying mechanism. So early life trauma is actually one of the, so Francesco asked this question about biological mechanisms for mental health. Early childhood trauma is the best exposure to trigger biological responses in an adult. So you see reduced telomere length, you see epigenetic modification, and so forth. So there are these long-lasting effects of poverty and early childhood trauma, and they are exactly in the pathways which we think air pollution and other environmental exposures are also promoting. So yes, I agree. I think it could be an addition. And one of the fascinating experiments from the United States is there are people who have exposed mice to air pollution and to stress, and actually pregnant mice. And actually when they did this at concentrations where there was no effect. But when they combined the two, actually the mice aborted the fetus. And so this is something which they do if their life is getting unpleasant. So indeed, there are these combinations, and I think we should really look into that more.
Okay. Right, thank you ever so much. And I'd like to thank all the speakers for this afternoon's session.